Hi there, and welcome to our part one of our lecture on gene expression. So in this part of our unit, we're going to be looking at how our genetic material, how our DNA is expressed, how we take pieces of our sections of our DNA called genes, and those genes will be transcribed into a messenger RNA molecule, and then from there they will be translated into a protein. So we are going to, this is referred to as gene expression. So how are these different genes expressed, and what do the cells do in order to express the material needed for our cells to function? So gene function or gene expression is gene function at the level of traits, right? So we have what's referred to as a phenotype. What we look like, what we see in another organism is its phenotype. But in addition to what is observed, right, in addition to what we observe is we also, get my pen working here, so we have our observations. So in addition to what we observe, this is also what our cells express. And what I mean by that are proteins. So gene expression is more than just what you see visually. It's also what proteins is a cell expressing. What proteins is a cell producing that is causing that cell to carry out its particular function. So a cardiac cell, it only expresses genes that create proteins that are responsible for proteins that carry out the functions of a cardiac cell. Same thing for a liver cell or a skin cell or a nerve cell. So all of these different cell types, even though they're all coming from, they're all yours and they're all your genome, different regions of that genome, of that DNA, is being expressed in different cells. So not only do we have what we see, such as a physical trait like skin color or hair color or eye color or height, what we also need to remember as a phenotype is an individual cell has a phenotype, and that individual cell's phenotype is its proteome, the collection of proteins that it is expressing, that it is using. Gene function at the molecular level is where we look at how a cell is actually functioning itself. What is its genetic makeup? So when we're talking about its genotype, what is the DNA sequence? Oh, that got erased. What is the DNA sequence that is responsible for the phenotype of an organism? So when we look at gene expression at the level of a trait, we're looking at a phenotype. We're looking at what we see and the proteins that are produced, the products of those genes. When we look at gene function on a molecular level, we're looking at the genotype of an organism, and that is the actual DNA sequence. And this is important because these genes come in different, what I like to call flavors, but they're called alleles. And alleles are different versions of the same gene. So we all have an allele for hair color, or we all have a gene for hair color. We just have different alleles of that gene. We have different versions of that gene, so that we all have different hair colors. Now, when we put the two levels together, both a genotype and a phenotype, we are now looking at the structure and function of a cell. And this structure and function is what determines what traits are going to be expressed. So our DNA is responsible for, the genes, I should say, within our DNA is responsible for the blueprint to create a protein. And those proteins carry out the functions of our cells. And the functions of our cells oftentimes are responsible for what we call the phenotype of an organism. So in order to understand how our genotype led to a phenotype, there were a couple of different people that were very important in this process. And they studied the mutations of genes in order to understand how the genes function. And to this day, that type of study is still done when we're not sure that we understand the function of a gene. So think of it this way. If we have a gene that we don't understand what it does, well, let's mutate it. If we remove the function of that gene, how does that change the cell? Now what happens to the cell? Does the cell shrivel up and die? Does the cell express proteins of a different level? Does it change the function of a cell? If we go in and we have a gene, but we don't know what that gene is responsible for, just by simply knocking out the gene, this is sometimes referred to as a knockout, by just knocking out the gene through mutation, we can observe what changes are occurring to the cell. That tells us, and then if we, of course, reinstill that gene back to its functional nature and the proper function of the cell comes back, we can then pretty definitively say, hey, this gene is responsible for this protein that carries out this function. And so now we understand, once we understand the role of that protein in the cell, we now understand the role of that gene in the overall function of the cell. So some genes are what we call constitutive. They are genes that must be used all of the time, such as the genes that encode for all of our enzymes in metabolism. But then we have genes that are non-constitutive, and non-constitutive genes are those that we express only when we need them. There's a few that we have that are only expressed during early embryonic development. We only use them at that time period for very rapid cell growth, and then they get turned off. And we don't want to turn those genes back on, so they're off permanently for the rest of your life. So by looking at the function of a gene, we do this by looking at its DNA, the sequence, of the DNA for that gene. So Archibald Garrod was one of the first people to start describing the relationship between a gene and the production of an enzyme. So the first proteins that were really studied were enzymes because those are functional proteins. Now, what Garrod did was Garrod studied patients that had defects in their metabolism. And this works out well for us since we just finished a chapter on metabolism. And he, in particular, studied a disease called alkaptonuria. In alkaptonuria, a person cannot break down a certain amino acid. Okay, they don't have the ability to break down a portion of an amino acid called homogenistic homogentisic acid. And in alkaptonuria, they accumulate abnormal levels of this homogentisic acid. And what happens with this acid, it's called alkaptin. And alkaptin will be excreted, the waste or the excess alkaptin will be excreted out of the body as a black precipitate. So it causes skin and cartilage to kind of discolor, so the skin gets a strange color to it. But the most disturbing side effect is that when these people urinate, their urine is black. So down at the bottom of this screen here, you can see we have urine from somebody with alkaptonuria. Now, what we're looking at is the breakdown of phenylalanine. So alkaptin, right? Alkaptin is an intermediate in the breakdown of an amino acid here called phenylalanine. So phenylalanine is broken down by phenylalanine hydroxylase into tyrosine. If a patient has a genetic mutation, in their gene for phenylalanine hydroxylase, then they have a disease known as phenylketonuria. In phenylketonuria, this enzyme does not work, so they have an excess buildup of phenylalanine. Now, this can become deadly to a patient. It can be a big problem, but it is easily controlled through diet. Now, some patients don't have a mutation at this particular enzyme, so their pathway will move forward. So when they take in dietary phenylalanine, it'll get broken down into tyrosine. Tyrosine gets broken down into hydroxyphenylpyruvic acid, and hydroxyphenylpyruvic acid will be broken down into homogentisic acid. But this process here, from the hydroxyphenylpyruvic acid to homogentisic acid, requires an enzyme called hydroxyphenylpyruvate oxidase. This enzyme here, if there's a mutation in the gene for it, doesn't work. If it doesn't work, then the patient gets a buildup of this hydroxyphenylpyruvic acid, and this is a disease called tyrosinosis, because tyrosinosis, it's basically excess tyrosine. Now, sometimes this does work. So if the patient doesn't have a a mutation in this enzyme, the last or final version of this disease is alkaptonuria. And this is where homogentistic acid oxidase, the enzyme that breaks down homogentistic acid, this particular enzyme has a mutation in its gene. So if this enzyme does not work, then homogentistic acid builds up in the cells because the patient has a disease known as alkaptonuria. So we have 3 different disease states that can occur here. I'm going to try to erase this if I can. Sometimes it will erase and sometimes no, it will not. So anyway, what I wanted to show you here was that alkaptonuria is the 3rd of 3 diseases that can occur in the same metabolic pathway. This is all the pathway to break down phenylalanine. It all just depends on where the mutation occurs. And the mutation occurs because DNA encodes for RNA, encodes for protein, and enzymes are proteins. So if my DNA is wonky or has some kind of mutation in it, my RNA will have a mutation, and when my RNA is translated, my protein will not be functional. Now remember, proteins are functional because of their 3D shape. And that 3D shape is determined by the primary sequence of a protein. And the primary sequence of a protein is determined by the DNA. So the gene for a specific protein says, hey, you need to put these amino acids together in this specific sequence. And it's in that specific sequence that that protein will fold, because the R groups, the functional groups and R groups of the amino acids that make up that polypeptide are going to interact with each other in secondary folding, and then in tertiary folding they're going to start interacting with farther away neighbors and develop their 3-dimensional shape. And the 3-dimensional shape of a protein is what determines its function. Now, what Garrett figured out here was this was all going on in this one pathway. And he came up with the idea that the diseases he was observing, particularly alkaptonuria, was because there was an enzyme missing in the pathway. Now, the enzyme wasn't really truly missing, but the enzyme's function was missing. So this enzyme may still be encoded for, it may still be produced, but it's being produced with a mutation in it, and that mutation is resulting in an enzyme that is not forming its proper 3-dimensional shape. And without its proper 3-dimensional shape, that enzyme will not have any function. Because this was an inherited pattern that he observed, he knew that if the patient inherited this trait, he had to get it from both parents. So he referred to this as an inborn error of metabolism. Now, this whole idea of inborn error of metabolism and this idea of looking at metabolic pathways to observe genetic mutations was really kind of catching on. And there was a team by the name of Beadle and Tatum, known as George Beadle and Edward Tatum. They were in California. They were at, I think, at Cal State or University of California, something like that. They were out in California. And they developed mutant strains of fungus. They were studying other organisms. They were actually studying, I think it was a beetle here, was working on fruit flies, on the eye color in fruit flies. But the fruit flies were difficult to deal with. So he and Tatum came up with the idea of observing these inborn errors of metabolism in fungus. And they started growing mutated strains of fungi, of mold. And what they did with this mold is they exposed it to X-rays. Now, they knew that X-rays, or what's known as irradiation, could induce genetic mutations. So they started irradiating or X-raying this fungus, and they would expose it to X-rays. And then they would grow the fungus on different types of media. Now, fungus grow on, it's called agar. It's kind of like a really hard jello. But you can add different nutrients and ingredients to this agar resource and provide specific nutrients to the organism growing on that agar. So in this case, they were providing very specific nutrient sources that coincided with enzymes that were in these particular pathways. So they started comparing what's known as a wild type. The wild type is kind of the normal expression of an organism, right? So they call that wild type. And then they started mutating this fungus. So they had, for a control, they had a wild type fungus. Then they exposed it so that there was a single mutation. And they knew this because it would require the addition of a single vitamin to the media. Now, normally, these precursor metabolites here would be given, and the organism would be able to break these down itself, and it would produce this arginine 4,7, and that would give us ornithine. But if this mutation, this gene here was mutated, then ornithine would not be developed. And in order for the organism to grow, Beetle and Tatum had to physically add ornithine to the media. Now see, if they just put the original precursors in, the fungus should be able to grow throughout this entire cycle. But some organisms with a single mutation couldn't even grow to begin with. So what Beetle and Tatum did was Beetle and Tatum took some of those that were dying originally and added them to a plate with ornithine in it. If they had a mutation in only this gene, then by adding ornithine, the fungus would be able to grow. In another set, they realized that they had what were called two mutations. So they took the organisms that couldn't grow without ornithine. Some of them could grow all the way as needed, but others could not get past citrulline. So they had a mutation in this gene as well as in the first gene. So they had a mutation in arginine 2,3. In this case, Beetle and Tatum had to add citrulline to the media in order to allow the organism to grow. And then if the organism had 3 mutations, meaning all 3 of these genes were not working, the only way Beetle and Tatum could get the organism to grow was to add arginine to the media. So these are, all of these are amino acid precursors. And Beetle and Tatum, they were just looking at these different mutations where they were occurring and how many of them. So this is known as the arginine synthesis pathway. Arginine is an amino acid that has to be synthesized. And it's thought to involve these precursor molecules, ornithine and citrulline. And so that's why Beetle and Tatum were adding these to the media to see what the different mutations were.
One of the geneticists who studied fruit flies alongside Morgan was George Beetle. Working in Paris in 1935, Beetle detected evidence that the inherited trait of eye color in Drosophila might be the result of genetically based chemical reactions. Beetle pursued his investigations at Stanford University with colleague Edward Tatum. For their experiments, Beetle and Tatum selected another simple organism, Neurospora crassa, bread mold. They chose mold because it was easy to grow with simple nutritional needs: bread, air, and water. Neurospora also had a single set of chromosomes, which allowed the researchers to observe genetic changes easily. Knowing that X-rays damage the chromosomes, Beetle and Tatum irradiated the mold, which caused mutations in the genes of its spores. These mutant genes were unable to produce the nutrients necessary for the mold to grow. However, when they added the nutrients, some of the spores began to germinate. For Beetle and Tatum, this was a breakthrough moment. They realized that the irradiated spores failed to produce the essential nutrients because their genes were defective. This was significant. It meant that genes were responsible for more than just passing inherited traits from one generation to the next. They also directed the production of enzymes that the mold depended on for its survival. Beetle and Tatum's breakthrough is known as the one gene, one enzyme hypothesis. Lactose intolerance is an example of a human metabolic condition caused by a single missing enzyme. Missing because some people have inherited a gene that fails to produce lactase and are unable to digest the sugar in dairy products. This disorder can be easily treated by taking tablets containing the missing enzyme. Thanks to Beetle and Tatum, we gained a fundamental understanding of what genes do, and the stage was set for a new generation of remarkable genetic discoveries.
So Beadle and Tatum really kind of introduced what we now call the one gene, well, it's now known as the one gene, one polypeptide, but originally it was referred to as the one gene, one enzyme hypothesis. And Beadle and Tatum really were able to show that genes encode for enzymes. That when the gene is not the proper sequence, the enzyme is not going to carry out its proper function. But what we now understand is that enzymes are only just one of many categories of different types of cellular proteins. Not every protein is an enzyme. So now, instead of saying it's one gene, one enzyme, we now say that it's one gene, one polypeptide. Because we also understand, in addition to that proteins are more than just enzymes, we also understand that some proteins have a quaternary structure. And in that quaternary structure, we have more than one polypeptide coming together with other polypeptide chains to create functional proteins. So not every protein is an enzyme. Not every protein is functional in its tertiary state. Some proteins must come together in a quaternary state, in a quaternary stage of folding, in order to become fully functional. Hemoglobin is our primary example of that. It is actually made up of 4 polypeptide chains all bound together. And hemoglobin will not carry out its proper function if all 4 of those chains are not together. So we have turned this into from one gene, one enzyme to a one gene, one polypeptide theory. So that brings us to the central dogma of biology. And the central dogma is that DNA makes RNA makes proteins. This is the central dogma. Everything in life revolves around this process. DNA to RNA to proteins. Now this process right here is known as transcription. And this process here, from RNA to proteins, is known as translation. And we're going to study transcription and translation. So in transcription, we are going to produce, our cells produce from our DNA sequence, they make a copy of the DNA sequence, but they make it in the form of RNA. So remember, DNA is a deoxyribose nucleotide, and RNA is a ribose nucleotide. So 2 different sugar molecules involved there. So we transcribe our DNA into RNA, and this is done and referred to as the transcript of a gene. Now, structural genes are genes that encode for polypeptides, for proteins. So structural genes produce a very specific type of RNA called messenger RNA, and it's written as a lowercase m and then all uppercase RNA. So when you see the term mRNA, you know that we are talking about an RNA transcript that is going to be responsible for being translated into a protein. In translation, this is the process of making the actual polypeptide itself. And the mRNA is going to be read. It's read by a ribosome, and the ribosome will translate that RNA message from DNA, will translate it into a protein message, into a sequence of amino acids. Eukaryotes, and then in prokaryotic organisms like bacteria, we're pretty much done there. The transcription and translation that occur to create these proteins, in bacteria or prokaryotes, it occurs simultaneously. It's not separated by any kind of membranes or anything. It's all occurring at the same time. And the genes of a bacterial cell or a prokaryote are very straightforward. It's the gene sequence is translated directly into an RNA sequence and translated directly into a protein. And so their central dogma is very, very simple. When we move into eukaryotes, it gets very complex. So eukaryotic transcription and translation involves multiple processes. We go through transcription and translation just like bacteria do, but we have an additional step, an intervening step known as RNA processing. And in RNA processing, the mRNA that comes directly off of the DNA molecule is not the same RNA, messenger RNA, that's going to be read by the ribosome. So we have to process our mRNA before it can leave the nucleus, and that's going to occur again in the nucleus. So when we look at molecular expression in prokaryotes, you can see we have transcription and translation all occurring at the same time. So here's our DNA molecule. We've got an mRNA molecule here. Here they're showing a ribosome. This is a ribosome. And that ribosome is in the process of translating this mRNA molecule. It's using another type of RNA over here called tRNA. And this blue little chain here is our protein. But when we move over into the eukaryotic cell, it's a little bit different. In the eukaryotic cell, we have our genetic material here, and it's transcribing into an mRNA transcript, but this is called a pre-mRNA. The pre-mRNA is going to be processed, and we'll talk about that processing in a little while. But it will get processed, and the final product of that mRNA processing is the final mRNA that will leave the nucleus, enter into the cytosol, and then be translated into a polypeptide. So here we have our final mRNA transcript, our ribosome, our tRNA, and then of course the protein product. Regardless of which, both went from DNA to RNA to a protein. So again, our central dogma is DNA to RNA to protein. Now, the genes that are located within our DNA are responsible for housing the information that's necessary to create us, right, to make us. So structurally, our structure is dependent on our genome, as well as allowing us to interact with the environment. My body needs to know if it's cold outside so that I know to cover it up and conserve my warmth. My body needs to know if it's overheated, so I can then expose my body to a cooler environment in order to cool it down, so that I can keep my body at its best functioning temperature. Structural genes are those that encode for polypeptides. We kind of covered that. There are genes referred to as non-structural. Non-structural genes encode for things like tRNA, ribosomal RNA, and RNases, which are enzymes that break down what they call ribozymes. And ribozymes are RNA molecules that have enzymatic activity, but they're not proteins. So structural genes encode for polypeptides or proteins. Non-structural genes encode for other things that are not proteins. Upwards of 80 to 90% of our genes are structural genes. So the majority of the genes we find in our genome are encoding for some kind of protein, which makes sense because proteins do all of the work. They are both structural and functional within our cells. So this polypeptide that is being encoded for by a gene becomes a unit of function. Okay, so that's the protein that we're looking at. And of course, what proteins do, what their particular functions are inside of a cell, determines the actual structure of the cell and what the cell as a whole can do. So the proteins that are being expressed or being produced in a cardiac cell are responsible for that cell acting like a cardiac cell. And the proteins that are being expressed and produced in a kidney cell are responsible for that kidney cell functioning as a kidney cell and not as a cardiac cell. So the traits and organisms of cells determine the traits and characteristics of an organism, and all of those characteristics are all based on the cellular activities that are encoded for in the genome. So Beadle was originally working on some eye color, and eye color he was looking at eye color in fruit flies. And the Drosophila, they're called, are one of the most well-studied organisms on the planet in terms of genetics. And the wild-type fly eye color is red. And so here we have the gene for eye color located on these two chromosomes. And when these genes are expressed, they cause proteins to produce these red pigments. Okay? So that's the function of those genes. So the mRNA, here they're walking you through the process, right? The normal gene gets transcribed into a functional mRNA molecule. That mRNA molecule gets translated into what's known as a transport protein. So that mRNA, right, this mRNA from the DNA is encoding for this transporter. Transporters are proteins, so there must be a gene for it somewhere. This transporter is responsible for pumping in precursor molecules. These precursor molecules, it's responsible for pumping it into the cell here. And when it gets pumped into the cell, it picks up these red pigments, and now the eye of the fly is nice and red. Over here, we have the same thing occurring, but we have a mutation in the gene. Notice the genes are on the same chromosomes in the same places, but this gene has some kind of change in its sequence. So that DNA sequence is different between the two. And because it has this mutation, it doesn't function. If it doesn't function, then the transporter we see over here is not there. If it's not there, then nothing can be transported into the cell, and so now the fly's eyes remain white. So transcription itself is when a gene is organized into a DNA sequence. And that DNA sequence in transcription gets transcribed into that RNA molecule, right? That RNA transcript. It's referred to as a transcript. Ultimately, that RNA is going to get translated by a ribosome and formed in the ribosome will use the blueprint of that mRNA to create a new polypeptide. Over 90% of all of our genes are structural, so I was a little off earlier, but you get the idea. The majority of our genes are encoding for proteins. And other genes encode for transfer RNA, like I said before, and ribosomal RNA, and something called a ribozyme. So this is kind of what our typical gene looks like, just to give you an idea of how these get transcribed. So here we have the actual gene itself, right? So this is the sequence of nucleotides that we want to transcribe into an RNA molecule. But in order for this gene to be transcribed, we have to know where it is, we have to know where to start, and we have to know where to end. So all genes have what's called a regulatory region, and that's what's occurring over here. Within this regulatory region, we have a promoter. A promoter is a sequence in this genome that very specific proteins are going to recognize. Think of the promoter as the mailbox to your house. If somebody wants to deliver a piece of mail to you, they have to put an address on it that's going to match the promoter so that that mail gets delivered to you. And they have to put your exact address on it. So a promoter acts like an address, and only the mail carrier or only the person who has that address knows to go there. So we are going to see sets of proteins called transcription factors. Transcription factors, and we have studied these actually. We looked at them in Unit 2. Transcription factors are proteins that are usually turned on by a cell signaling pathway. Remember when we talked about the RAS-RAF-MEK pathway, and we talked about the EGFR pathway and the GPCR pathway? Well, it was particularly in that the epidermal growth factor receptor. When we talked about that EGFR pathway and that kinase cascade, and eventually all of that phosphorylation was leading to the activation of a transcription factor. That transcription factor is the protein that is responsible for finding the specific promoter sequence of a gene. And that's how we regulate how a gene gets turned on and how a gene gets turned off. Sometimes genes are always on, and so we need to turn it off, and the transcription factor is responsible for that. Other times a gene is always off, and we need to turn the gene on, and a transcription factor may be responsible for that as well. So the promoter sequence is the, hey dude, I'm over here. It's kind of the big red flag telling its specific transcription factor where it is located so that transcription factor can get started on getting this gene expressed. Next to the promoter is the regulatory sequence, and this regulatory sequence works with the transcription factor as well as RNA polymerase and some other enzymes to kind of bring everybody together to get started on this transcription process. Once everybody is together and they get the go-ahead, transcription will begin. And in this case, this whole region here will be transcribed into an RNA molecule. And there's an enzyme that does that called RNA polymerase. And as that enzyme moves across, it reads the DNA sequence until it reads a sequence known as a terminator sequence. When it reads this terminator sequence, it's a very unique sequence, then the RNA polymerase molecule will fall off of the gene and transcription will no longer take place. And what's left is the RNA transcript. So the promoter is the site for RNA polymerase to bind and begin, right, signal the beginning of transcription. In the regulatory sequence, we get regulatory proteins. These are the guys who say yay or nay, right? This is where permission can be given or taken away. And then, right, so we see our promoter sequence, and here's our regulatory sequence. Then we have the transcribed region. That's the actual information that we want. That's the information we want to make our protein. And then finally, the termination sequence over here. The terminator is where we look and signal to our polymerase that transcription must end. So this is just lecture 1. This is a 3-part lecture, and I want you to think about these questions. And these questions are all relevant to what we've talked about just today. So what is a gene? What is gene expression? So how is a gene expressed? What do we mean when we say gene expression? What's the difference between structural and non-structural genes? What's the central dogma of biology? When you describe transcription, I want you to try and write it a sentence or try to describe transcription to somebody in terms of genetic expression. So talking about what role does the mechanism of transcription play in genetic expression? And how is the DNA sequence of an organism related to the actual physical attributes of that organism? What does our DNA have to do? How do we go from DNA to blue eyes, or DNA to dark skin, or DNA to 6'3"? How does the human body do that? What processes are involved? So those are some questions I want you to think about on this last lecture. They're things that you'll be asked about on the exam. So this is certainly a way to kind of look at our lecture in a bite-sized piece. Right? I will see you guys on the flip side in part 2.